Hello everyone, it's fantastic to be here. Welcome to my home, which is on the traditional land of the people from the Eora nation, and I would like to acknowledge the elders past, present and emerging. Today, I would like to talk to you about cloud-based bioinformatics and how to accelerate disease gene discovery and diagnosis. Now, events like SheBuild would not be possible without the female tech heroes of the past, who paved our way so that females can be at the cutting edge and stand on the big stage. One of my tech heroes was Ruby Payne-Scott, who was the first female radio astronomer at CSIRO. Now, Ruby was asked to leave CSIRO when she got married. She refused though, and went on to have a stellar career and made really important discoveries. Now, Ruby really changed the organization for the better, and what a fantastic organization it has become. So I would like to say thank you, Ruby, for your efforts. CSIRO is Australia's government research agency, and at CSIRO we're very passionate about translating research into products that people can use in their everyday lives. For example, one of CSIRO's most famous products is Wi-Fi, which CSIRO invented together with Macquarie University and is now used in over 5 billion devices worldwide. CSIRO also invented the first influenza treatment, Relenza, together with Monash University. And on a lighter note, CSIRO developed the Total Wellbeing Diet, which is now on the book bestseller list alongside Harry Potter and the Da Vinci Code. So I think from that perspective, we have a nice balance between products that people enjoy and products that people need. Now, my research is probably more on the needs basis, in that I'm part of the eHealth Research Program, which is the largest digital health initiative here in Australia, and worldwide quite unique in covering the full value chain from basic research all the way up to bringing service and technologies into the clinical practice. Today though, I want to tell you about 3 stories. The first one is around how we're using machine learning to understand the genome. The second one is how we're using serverless compute to edit the genome. And the last one is around better collaborations and how to do research faster using the cloud. So for the first one, I picked a quote that really resonated with me. It says, "AI is the new electricity." And this really speaks to me because AI or machine learning has become so ubiquitous that every industry and organization is using it. So from that perspective, it's no surprise that we applied it to genome medicine. Specifically, we're part of the largest health consortium looking into a specific disease called motor neuron disease, or ALS, which is the disease that Stephen Hawking suffered from. In this consortium, the aim is to find disease genes or the molecular origin of this disease. And our part in that is to compare healthy individuals with people that suffer from ALS in order to find the single genetic change or the set of genetic changes that contribute to people developing ALS. The reason this is difficult is because there's lots of data actually involved. And it needs to be in order for us to really understand this complex disease. So Frost and Sullivan was saying that by 2025, 50% of the world's population will have been sequenced. That means genomics will produce more data than the typical big data disciplines, YouTube, astronomy, and Twitter combined. Now, on one hand, this is exciting because for the first time, we're going to have the amount of data we need in order to understand a complex disease like ALS. But on the other hand, it's quite scary because as health researchers, we're not used to being thrust at the forefront of IT development. But as researchers, that's exactly what you do. You take on a challenge, you adapt, and you innovate. We've done that with VariantSpark. So VariantSpark is our machine learning technology or library that is able to analyze large volumes of data. We showed that it is 3.6 times faster than the traditional approaches that are out there developed by Google and Yahoo. And I'm showing here in this graph, I'm plotting the runtime against dataset increases. And what you can see is VariantSpark is the dashed blue line and the yellow line is growing sublinearly with the dataset sizes, whereas other technologies grow exponentially, meaning they cannot scale to the dataset sizes of the future. And we showed that in that dataset sizes of the future would take traditional approaches 100,000 years to process, whereas VariantSpark with its new algorithm can process that kind of data in 15 hours. So applying this power to the ALS problem, we were able to confirm existing disease genes, but also we were able to look into areas to join ancestry information. So bring family units that are previously thought to be unrelated together. And that brings more power to the analysis, because we know that the disease that is shared between the family unit is likely the disease gene. So with this additional power, we were able to identify 2 additional disease genes, which ultimately, hopefully improve the diagnosis. And this is important because in ALS, time from diagnosis to death is only 2 years. So people need to get diagnosed fast and get on a treatment as soon as possible. So to make this analysis available to researchers internationally, we put it on the AWS marketplace. This brings the benefit of researchers on the other side of the world being able to download not only the code, but the exact environment and interactions and data handling procedures in order to analyze their data safely. And more importantly, they can produce that kind of analysis on the data themselves that will never leave their account. So VariantSpark comes into their account, gets analyzed by the researchers themselves, and the result will stay in their account. So everything is under full control of the user, or the researcher in that case, without ever leaving the organization. The way we achieve this is by infrastructure as code. So everything from the compute environment, from the permissions, from the data handling, and the code itself is implemented in a Terraform template. This Terraform template is what is on the marketplace, and it can be handed over to other researchers who can then click a button and it automatically gets deployed into their account. Now this brings us to the second story around serverless compute and how to edit the genome. The quote that I chose for this segment is from Jennifer Doudna, who very topically won the Nobel Prize in Chemistry along with Emmanuel Charpentier. She says, the world around us is revolutionized by CRISPR, which is a genome editing technology. And she then goes on to say whether we're ready for it or not. And I think this is very telling in that technologically there will be a lot possible. But from an ethical and societal perspective, we need to think about whether those applications is something that we want. Now, something that we definitely want is to treat genetic diseases. And here we're working together with the Children's Medical Research Institute in Sydney, and they're interested in a genetic disease that attacks the liver. So children that are born with this misspelling in their genome will then develop a disease that attacks the liver and unfortunately dies from it. So therefore this treatment would be a life-saving treatment and will really be a game changer in that space. So the idea here is that the CRISPR technology is going to a specific tissue, in this case liver, and will correct this misspelling in the genome, ultimately forming a treatment and potentially a cure for that genetic disease. The reason though this again is difficult is because this is hugely computationally intensive, because the genome is a large space with 3 billion letters. This particular editing machinery needs to go to a specific location and need to go there very faithfully to edit only this gene and not other genes. So we need to identify not only that location but compare it to the rest of the genome in order to detect whether it is a good binding site or not. So this kind of computation is usually done in a traditional approach and would take a long time to compute. Whereas we brought it down to, or we use serverless technology in order to massively parallelize it and be able to improve and increase the speed, improve the accuracy and increase the speed. Now, the kind of cost saving that you can actually do with this serverless approach is visualized here with this application, where traditionally researchers will have to spend $4,000 per month in order to run a genetics or a genomic service. But we, using serverless technology, can bring it down to just $15. So for less than a cup of coffee per day, researchers can contribute to global knowledge gain. So what is really involved in doing genetic surgery? Well, first of all, you need to identify the misspelling in the genome that you need to correct. You need to identify which tissue is involved or affected. And then you do need to do the computational analysis in order to identify the exact location that you want to treat and package up the machinery in order to be delivered to only that particular tissue. Then you need to go on and identify how to make this generalizable. So you might have developed it for one genome, but you do need to apply it to other genomes so that more people can benefit from the treatment. And therefore, the changes that are in the general population, the genetic changes, there are on average 2 million differences between you and the next person. All of those must not affect the genome editing machinery in its task of going to a specific gene. Of course, from there it needs to go to preclinical trials and then clinical trials, phase 1, 2, and 3, before it can be rolled out as a general treatment. Now this brings us to the third area, because neither genome engineering nor genome analysis would be possible by just one research group. It needs global collaboration to really exchange the kind of knowledge that is necessary in order to do those leapfrogging changes in our procedures. So for us, being able to globally interact with other researchers means going to the cloud. And I could not find a quote that quite encapsulates what cloud means to us, so I made my own. Cloud for us means it's creating a more collaborative way of generating knowledge and doing business. So it's not a technology, it's a mindset. Yes, of course, it's just cost-effective scaling to variable workloads, as I highlighted with the serverless approaches where we can save 99% of the compute cost. It also lets us access the latest in compute technology without upfront investment, which I showed with Variant Spark, where we're using a Spark cluster without having to invest in a Spark cluster ourselves. But the most important thing for us with the cloud is really the connectivity to the world and doing reproducible research like never before. Being able to write a paper, writing it up as a CloudFormation template or a Terraform template, and publishing that alongside the academic text really enables other people to replicate and reproduce the analysis that we developed. Being able to collaborate with the world has never been more important than now with coronavirus and COVID-19. So here, using cloud and international connections, we're able to do analysis that just a couple of years ago would have taken forever and large consortium to actually organize and set up and do. For example, we analyzed whether the mutations in COVID-19 virus is actually causing a different kind of disease. Here we were able to work together with GIS8, which is the largest database holding viral genomes, and be able to identify 500 individuals to contribute to this study. 300 who had severe forms of disease and 200 who had a mild form of the disease. From there, we used Variant Spark in order to pinpoint whether there is a specific mutation in the viral genome that can cause one versus the other outcome in the disease progression. Now, interestingly, we found that there are a couple of locations in the viral genome associated with the outcome, and one of which is the well-known D614G mutation, which is in the spike protein and has already been associated with a more virulent version of the disease. But this kind of analysis is unfortunately underpowered, in that the genome of the virus is 30,000 bases, and we only have 500 individuals in order to identify which ones of those is associated, which is clearly not statistically significant. So therefore we need to wait until there's more data accumulated, better annotations for those samples, in order for us to redo this analysis and really answer the question whether the virus can mutate in a meaningful way to have clinically different clinical outcome. Another area where we use technology in the fight against COVID is around diagnosis. Here, people around the world are working on a pregnancy test style approach for doing diagnosis rapidly and outside the lab. The idea is that CRISPR technology can be used in order to target a specific location in the coronavirus, in this specific strain of the coronavirus, and would detect that. And that detection can then be used in order to differentiate whether someone has the disease or not. Now, this detection and CRISPR diagnostics in general is a programmable approach where a certain element can be swapped out to recognize other forms of the virus, which means that the programmable element can be optimized in order to better detect, so reduce the false negatives, and strongly differentiate between different strains of the virus, so potentially reduce false positives. And this is exactly what we've developed with Sorte, a computational tool, cloud-based, serverless, that is able to optimize the programmable element in CRISPR diagnostics kit to make the approach more accurate and more sensitive. So the three things I hope you took away from the talk today is that science is accelerating through technology. We constantly need to reinvent ourselves. But greater capability also means greater responsibility, in that we really need to look at whether the technology that we're dealing with is actually something that we want. And the last one is that technology, IT, research, science is generally becoming more collaborative. So industry and technology and academia should really work together to achieve more. So this brings us to my last final thought here in wrapping up with Ruby, in that Ruby single-handedly revolutionized the organization, but I think each one of us, in doing our individual changes, like being on stage, like presenting, like being visible at the forefront of technology, will together enable us to make heroic changes going forward. And let me wrap up this talk by thanking the fantastic team that I am working with. And let me refer you to my Twitter account, where I'm sharing our research findings and other news. For example, the program has recently released 2 reports, one around how we're using AI in the health sector, listing 32 case studies of how machine learning and AI is applied effectively in order to improve healthcare and health services. And the other one is our annual report, showcasing what the 100+ researchers and engineers working for the program, how they're contributing to Australia's healthcare system and improving the technologies going forward. With that, I'm looking forward to the Q&A session, and I'll see you there.